All right. Hello, everyone. Today we're going to be talking about mutation rates. So put on your thinking cap because today is going to get rough. So, without further ado, sorry, I've been sick. I might sound weird, but I'm going to do the best I can, okay? All right. So what you're looking at is a chart showing that pedigree mutation rates are very different than the evolutionary assumption phylogenetic rate, which based on the fossil record and split divergent time of common ancestors. Usually when we're referring to this, we use the chimpanzee split 7.5 million years ago in human beings. Because we're mostly going to be focusing on human beings in this, that's important to know. Now, I am going to touch on some animals to show that they also agree with the pedigree mutation rates, which is the young Earth creation results. But we're going to be focusing mostly on humans because these types of studies have been done many more times than animals have ever been. So to go over and to reiterate really quick, molecular clocks, all of them are calibrated to the reference of human chimpanzee split, which was assumed to be between 6 to 7 million years ago. So they build their mutation rate based on this assumption being true. We don't really like that too much because we believe that what we observe in studies is more plausible than what should be just assumed based on random chance of some theory, right? So when we see a mutation rate that's very quick, and it doesn't line up with the fossil record, that's what we would expect as creationists because the fossil record is not true. So what do these dates point to? What are they showing us? Is there any consistency, or is there a theme, or are the dates really just all over the place and there is no such thing as a clock? Now we're going to cover that today. And another thing we're going to cover is the difference between substitution rates and regular mutation rates. So what that means is that substitution rates deal with populations. So because the evolutionists like to harp on saying, well, you're not looking at the substitution rate, you only look at the mutation rate, and that's not right. Well, they kind of work out to be about the same anyway. But to appease them, today we're going to be looking at studies that looked at the substitution rate and show that they also agree with the fast mutation rate. So I would have liked to have been able to find a nice visual for the Hall study in 1995, but I could not do that. So we're going to skip over that one and jump into this study that was done the very next year in 1996. And it was done by Kate Bendall in 1996, who obtained 4 substitutions out of 360, or 1 out of 85. But this is looking at the control region and a specific region inside of it called the HVR1. So I highlighted it right here. You usually want to look at both. That's the goal of any type of study that's going to actually be looking for an accurate substitution rate, because HVR1 and HVR2 tick at different rates. HVR1 ticks slower than 2. So you take the 2 and you combine them, and right in the middle is your accurate rate. So you're always going to get a slower substitution rate by looking at this one. And you can see that is done here. You'll notice that it's 1 substitution out of every 85 generations. Parsons was around 1 out of every 30 to 33, which we'll get to. So this is twice as slow as the next studies that we'll see. They do get a little bit better, but you'll notice that this one right here is the next study done in 1997 by Mumm. Interesting name. HVR1 was the only region that was tested. You'll notice that got pretty good results. 1 out of every 55 generations found a substitution. These studies were not designed or intended to investigate a mutation rate in mtDNA. Rather, it was to verify 2 families that were related to one another. So they did not need to test the entire D-loop region. They only had to do one of the regions, and it wasn't to try to obtain an accurate substitution rate. They were trying to find relation. That's why they didn't waste their time with HVR1 and HVR2. They didn't need to. Also, Mumm, the study that came right after that, they only looked in one region. Again, that collects only half the mutations that occur in those regions. So, which ended up overlooking different heteroplasmic mutations, and it explains why they got a slower result. But then the next study comes, the largest ever yet of its time in 1997, obtained by Parsons. He noticed that there were 10 mutations per 327 generations. Again, these are substitution, which works out to be 1 every 32.7, which was the math that was done on the last one. He also made sure to not include the poly-C tract, which can skew up results. And you'll notice right here, he calculated both of the regions. That's why his results were so good and so much more accurate, and why they're used by the FBI to this day. You'll see in the report that the mutation rate also concurred with the substitution rate, and we landed on a mitochondrial eve around 6,500 years ago. And his results are so good that they've been published in the Forensic DNA Application Analysis Handbook, and they are used by the FBI to track down killers. So his results are very accurate, or else we would be convicting the wrong people.
And yes, they reported this in their initial study. I highlighted that area for you. Next, I think there are people out there who doubt this, or think that we're lying in regards to the results of these studies landing on 6,500 years date for Eve. So I will show you here directly from the study itself, that is what the results were. And I'll just read part of this here, starting at the first red arrow. I'm just going to read word for word so you can see for yourself that it's not just some made-up young Earth creation argument. Using our empirical rate to calculate the mtDNA molecular clock would result in an age of mtDNA most recent common ancestor of only 6,500 years, clearly incompatible with the known age of modern humans. Even acknowledging that the most recent common ancestor of mtDNA may be younger than the most recent common ancestor of modern humans, it remains implausible to explain the known geographic distribution of mtDNA sequence variation by human migration that occurred only in the last 6,500 years. So as you can see, they can't explain it, but that's what the results were.
Next, they didn't believe it, so somebody joined his team named Holland, who came to join him to refute his work and actually confirmed it, but actually even made the rate faster, because they actually discovered that there were 10 substitutions every 306 generations, which would make that 1 out of every 30 generations, confirming the fast mutation rate yet again. So fast mutation rate, fast substitution rate, they both land on the biblical time frame. Of course, some people would be like 6,500, that's 500 years off, you know, if they want to really get petty. But that doesn't matter, because we also have the Greek Septuagint, and which would say that actually Adam and Eve were around 7,500 years ago. But I guarantee you, you give that to them, they'll just discard that anyway. They don't want answers. They're not interested in these kind of facts. They're just interested in you being wrong. Just remember that. Next. The next study that was done, she found 1 substitution every 61 generations, right? Which is close to the previous ones. So then we have another fast mutation rate. Why aren't these landing on evolutionary deep time frames, 150 to 200,000 years ago? Because those dates are made up. They're not true. That's why they invented this phylogenetic method, because these dates confirm that it's not good for evolution. The next one, how at all? He comes forward and he does another one, but he pooled his information together. He used the control region, and he pooled data together from all of these other studies. Some of them found no mutations at all. But guess what he obtained? 2 substitutions every 88 generations, which works out to be 1 out of every 44. Very close to Parsons and Holland anyway, even though they were pooling the data together with other studies that were not generally as good. So again, what is his conclusions on this? In 2003, he stated, as in other studies, our estimation is much higher than those obtained by phylogenetic methods. See, they're starting to realize, wait a minute, every time we do one of these observable studies, it doesn't match. You see, the critics will say things like, oh, well, maybe they're counting somatic mutations, like these experts don't know what they're doing, right? Or, oh, in the deep past, selection was a lot stronger. You'll notice that they just throw out stories without any evidence, without any facts, with nothing, because they need their story to be true. And these studies prove that their story is not true. That's why they hate these mutation rates. There's other reasons they hate them too. But I digress. Next one, Sergren, I'm not even going to try to begin to pronounce his last name here, came to another study, and guess what he found? Both of our studies, his and Parsons, came to the remarkably similar conclusion. They state that right down here. Another one, extremely fast mutation rate, came to the same exact thing. The rate of mutation, 1 substitution every 25 to 40 generations. Oh, Amazing. Exactly the same thing yet again. It's a concurrent theme, isn't it? So as discussed by Sergrid, remember, in 2000, it is rather a phylogenetic rate estimate that it may be biased. Interesting. Now he's saying their evolutionary rate is biased because it doesn't ever agree with what we see. Amazing, right? It is clear that the pedigree studies prove a more direct estimate of the mutation rate. Are they? Can I dare say they're actually onto something here? How about the next one? In 2005, Santos here, this study looked at the entire D-loop region, very similar to how Dr. Nathaniel Jeanson did in his 2015 Ding et al. Study, and they found 11 substitutions out of 321 generations, which is one substitution every 29 generations, basically the exact same as Parsons, who got 1 out of 30 and 1 out of 33. Amazing, right? Constantly finding the same results. Then in 2012, we're getting more current. Basically found the identical mutation rate as Parsons did. Look at that. And here in the highlighted section, it says these values are much higher than those even estimated by Howell's estimations, and 13 to 42 times higher than the phylogenetic estimates for HVR1 in the same range. So imagine that, right? The phylogenetic rate, based on evolution being true, assumes, well, what do they assume? Let's look. They predicted that there would be one substitution every 6,000 to 12,000 years. But that would not even be close to it. One, even in our entire lifetime. That means that one substitution would occur at how old the Earth is. Yet we're seeing 2 or 3 substitutions occurring in a human lifetime, if it's every 30 years. So very easy for us to explain. And theirs is based on the unobserved guessing of the fossil record. So when we see these admissions here, that it's 13 to 42 times higher than their estimates of that particular region based on the fossil record, which is also a slower region, as you guys saw earlier, then what does that tell you about the faster region of HVR2? And it's just that much more devastating to them. How about this one? They did a pedigree study on this one, on the haplogroup, and they believe that this haplogroup formed about 4,000 years ago. Oh, the ancestor to that haplogroup is just 4,000 years ago. A whole people group. Interesting, right? So we're going to get more into that one when I'll move that even further down so we can cover it in a minute. Here's a model that was created, a simulation based on the data that they had. They pooled everything together up until this point in 2004. And they said that the MCRA, most recent common ancestor of all present-day human beings, lived just a few thousand years ago in all of their models. So every time they ran it, they could only get it back a few thousand years, exactly what we would expect in the biblical time frame, in the biblical model. Even simulations agree with it. So evolution theory estimated that one substitution every 300 to 600 generations to go from this to this. That is why they need to slow these rates down. It is devastating to them. So that study that I had just showed you, I believe it was this one. Yes, the simulation. This was their result on their time frame of that. And you'll see that the most recent common ancestor was about 300 BC, and all modern human beings 3000 BC. Oh my goodness, look at that. What a shocker. What else do we have? We have another study here that was done on demographics. It says the more realistic model estimates that the most recent common ancestor of mankind lived as recently as 3,000 years ago, and that the identical ancestor point to a recent several thousand years ago. Look at that every single time. So what do we find? We're seeing constant, uncalibrated mutation rates based on no assumptions of the past, telling us that the mitochondrial clock is showing a most recent common ancestor of Eve living just thousands of years ago, not even tens of thousands, let alone hundreds of thousands or even millions of years ago. None of that's true. That's why this works. And look at this statement here. Using the new clock, she would estimate a mere 6,000 years ago for Eve, the mother of all living beings. Interesting, right? Now I'm going to read something that comes directly from my book on mutation rates. And it is, how do we explain the diversity we see in the biblical timeline? Well, we explain it by looking at these observable mutation rates, and we can easily do that. The evolutionary model can't explain the diversity of all life using these mutation rates. That's why they discard them, they don't look at them, they don't like them, and they use their own made-up model anyway. So how would we explain slower mutation rates, or the fact that some of them actually don't show any mutations at all? Does that mean we can just throw away all of these other results that we're getting? Well, there's reasons for some of these discrepancies. We'll go over a couple of them now. One is that studies obtain slower mutation rates by looking at only half of the hypervariable regions in the D loop. Or even worse, they only investigate, they don't even look at the entire region, or they look at just half of the HVR1 or HVR2 sequences, right? So you're only going to get half the results. Studies only looked at related populations or in small groups, sizes with little to no diversity to compare the different rates to. That did not go very far back in time as well. This can work for a mutation rate, but it will never work for a substitution rate because you need different demographics to obtain a good substitution rate because you're looking at populations. Number 3, studies either looked at identical twins, single family lineages, or isolated and inbred families. These studies are prone for error, and since mutations don't show up at a rate of 1 per generation, they're much less than that, it takes a few generations to even see a full mutation rise. This is why you need a larger pedigree, or a deeper rooted one, so to speak, and why the larger pedigree studies and faster mutation rates give these types of results. Number 4, heteroplasmy was ignored. Heteroplasmy is a term that's used for different types of mitochondria in the same person. They always thought that we only had one type. Then we found that there were actually other types. You get faster results as well. So they're oftentimes ignored because you're just increasing and making the problem compoundingly worse for evolution by doing it. Number 5, mutation rates have never been observed to change dramatically, and testable predictions can be made and are being made by young Earth creation scientists and apologists. This is why a lot of these types of studies now are so taboo for the evolutionist, and they like to tag things on them like, oh, no, we don't agree with the results from these studies. Of course you don't. So, number 6, adding simulations into studies trying to make phylogenetic rates converge with one another. That obviously slows the rates down because they're mixing myth and fable with actual facts. Number 7, rule out any mutation they deem to be somatic because somatic mutations aren't passed on, right? So this will slow the rate down again dramatically. Number 8, data is often pooled from studies that have not been focusing on obtaining accurate substitution rates, and the studies where no mutations were found, and this obviously slows down the overall mutation rate dramatically, as we saw with a couple of studies already. Number 9, use a higher generation time to push the dates back further. You see, if humans are having a generation time around 40 to 50 years old, that's dramatically different after 300 generations than a family that's having generation time at 20 years old. So they just add higher ages by helping them push things back in time. And finally, number 10, trying to say that mutations occurring in the coding regions are accurate over a short period of time, but not accurate over deep time. They admit that mutations that are not lethal in the mitochondria can persist, but are negatively selected to the host. And over hundreds of generations, these will persist and give accurate clocks. But then they turn around and say, over thousands of years, they will not be accurate because these mutations will be removed by selection. So they invoke deep time and these kinds of assumptions as a rescue device. And you'll notice that they're conflicting. They literally refute one another when you listen to that argument. So that is the mindset that we're dealing with. They'll say something, even if it refutes themselves, just to prove you wrong. That's the kind of mindset that we're up against today. It is pretty clear that pedigree mutation studies provide a better direct estimate of that. So the critics don't really like Nathaniel Jensen. They'll say, well, he didn't consider coalescence. Look, from his very own study, this represents a coalescence calculation, the calculation most generous to the evolutionary model. So I'm sorry, yet again, evolutionist, you are wrong. Coalescence was considered, and it's considered in all of these other studies that we've been looking at as well. Another one, they said, oh, well, they're probably counting somatic mutations. Well, guess what? They thought of that a long time ago because this was early on in these Parsons studies in the 1990s and early 2000s. And listen to what they say. Our data clearly show that the heteroplasmic mutation in their lineages were inherited through multiple generations, and therefore they cannot be somatic. They can't be. Here, somatic variant is untenable. It's not even a good argument anymore, is what they're saying. So remember, they're trying to say that we're counting mutations that aren't being inherited, called somatic mutations, and that is why we're getting such a fast clock. They're admitting that that is not the case and that is not true. Even one of the biggest critics of Nathaniel Jensen's work has stated when you compare the two different regions, the D loop and the mutation rate of the entire mitochondria, and you line them up from his own study, the Ding et al., that it is true that these approximately match. So you can't just say, oh, it's just Nathaniel Jensen's results that's skewing everything. It's just another creationist lying to you when they match the secular studies that aren't young earth creation oriented whatsoever. All of these studies, the people believe in evolution, and yet they're getting young earth creation results. So the coalescence agrees with exactly what we would expect because, as you can tell, we're using Mendel's accountant on our model. And Mendel's accountant takes into consideration these variants. And when the NIH, which is the National Institute of Health, when they want to use a simulation, what are they using? They're not using some evolutionary assumption-based model. They're using the young earth creation simulator right here, which shows that deep time cannot be true because of the waiting time problem. And it shows how rapidly mutation and cancer spreads through a population. Why aren't they using some evolutionary model? Because it's not true. Listen what they wrote. Mendel's accountant can simulate with unprecedented biological accuracy the results of this accumulation. The simulation has shown the extinction of the human race only after 350 generations. If these result is anywhere close to correct, that humanity's genetic mutations would have led to extinction within 350 generations, how could it possibly fit with the evolutionary timeline? It can't. That's what they're running into, is into a wall. And when we look at how long it takes for beneficial mutations to become fixed, this is called the waiting time problem. You can see on the very chart, you're waiting 84 million years for just 2 mutations to be fixed. It can't be possible, everyone. So coalescence is taken into a consideration. Now, look at all these studies right here. I made this chart nice and easy for everybody to see. Mutation rates and substitution rates over here. Going all the way back to the early 90s, all the way up to 2015. We have a range for mitochondrial Eve living between 4,000 and 10,000 years ago. Nothing even remotely close going back to a bottleneck 200,000 years ago. So that's why they invent their stories, right? So we have the substitution rate right over here on the side. It's a nice little picture cheat sheet for everybody. But what about Neanderthal and their substitutions? Well, let's think about that for a minute. Look at this. We observed a similar number in Neanderthal, which was 20, and humans 18. So they only have 2 more than us. So what's going on? How could they have just 2 more than us? Because what would be the average in a Neanderthal? Look at that. We find 8 substitutions plus or minus 3. That's the average amount of substitutions. But wait a minute. They tell us that Neanderthals lived for 400,000 years and humans have only existed 200,000 years. Do you see the problem with this? That means that human beings living in a population for 150,000 years before we broke out of Africa, we were getting substitutions up to 20, and then we kind of plateaued, right? What do we see with Neanderthal? Well, how in the world could it possibly be true then that Neanderthal have fewer substitutions than humans when they lived in a smaller population of just under 20 for 400,000 years? They should have a minimum, a bare minimum of 40 at least. And we're talking they have a much smaller size. Now, remember, a population substitution fixation is based on population size. What's going to take longer for one of these people to walk to the top of this pyramid or to walk to the top of the smaller one? Obviously, it's going to take a lot shorter of a time to reach the top of this one. Well, that's how fixation works. You get a mutation. If the population is small, only, let's say, 15 people here, well, it doesn't take long for it to spread through the population until everybody has a fixed mutation in that region. That's how substitution works. We can explain this from Adam and Eve going to the Noble Flood bottleneck. Then there's a small population because people were all living together until the Tower of Babel. Then they disperse. So there's a good 1,000 years at least of rapid substitutions reaching fixations in populations and people groups, which we're going to get into in a minute. But this is better explained from our model about exactly what's going on and how fast and readily it can happen in a population. I cover all of this in the book, The True Mutation Rates. So let's look at a chart that was made by Howell in 2003, I believe. He said, this is what we observe, and we don't find anything in between, but yet this is what is estimated based on evolution being true. What's going on here? Why is this here? What's the problem? Can anyone solve this? Besides assumptions, nobody's been able to resolve it whatsoever because you need evidence to resolve something. They throw out hypotheses and stories all the time, but this problem still exists. What we observe is the exact opposite of what is estimated to be true, and they can't figure out why. And it's so simple. The story is not true. That's why. It's really just that simple, guys. This one says it all right here. We find that the mutation rate is approximately consistent per year and largely similar among genes. Ooh, look at that. I dare say, are they admitting something here? Look at this. Now we read what the study states about the lowest rates they obtained. You will notice that even the slowest rate they can honestly allow for is twice as fast as the common phylogenetic estimates. So based on pedigree studies, under the most conservative assumptions, we can conclude that HVR1 mutation rate from the Costa Rican population, which is about a 95% probability, fall between 2.75 times 3. Oh, look at that fast substitution rate there. In other words, the lower limit of our estimate is approximately twice as high as the most commonly accepted phylogenetic estimate. Isn't that unbelievable? Their slowest rate, they can't even possibly get that close to it. I like this one right here. This was written by Ann Gibbons. Of course, she wrote at the very ending what we read earlier. Eve would be a mere 6,000 years old with what they found from these clocks. Now, here's the funny part. This is what's concerning them. For example, researchers have calculated that mitochondrial Eve, the woman whose mtDNA was ancestral to all living people, lived 100,000 years ago to 200,000 years ago in Africa. Using the new clock, she would be a mere 6,000 years old. Now, listen, pay attention. No one thinks that's the case, right? Because they believe in evolution. But at what point should our models switch from one mtDNA time zone to another? I'm worried. Yes, you should be worried. You should all be worried if you believe in evolution, because you're believing in a fairy tale. That's why you're throwing out selection and all these other random things in hopes and in dreams that you're still related to a sponge. Next, the evolutionary dates are clearly dependent on many tenuous assumptions. Oh my gosh, they admitted that. Really? What else are they going to admit? Uh-oh. There's a general perception that there is a competition to get the oldest date, and there's kudos in it. Oh, could it be if they can prove evolution, they're actually going to get raises and benefits and more notoriety and be in more magazines? You bet they are. And that's their incentive and that's their drive. It's not about truth. It's not about evidence. It's about proving their fable correct so they can get more grant money and pat each other on the back. That's what's going on here. So reading the entrails of chickens, guess what they found on their molecular clock when they were looking at it, trying to say, let's view this from our evolutionary advancement. Despite their allure, we must sadly conclude that all divergence estimates, divergence from a common ancestor, discussed here are without merit. Our advice to the reader is, whenever you see a time estimate in the evolutionary literature, demand uncertainty. Do people ever demand uncertainty that believe in evolution? Of course they don't. They're immediately defensive against it. Matter of fact, they get mad at you when you tell them they were lied to, and then they go and defend the liar. That's how crazy they are. So they're being even told, like, to demand uncertainty. There's no truth to any of this. What's going on here? It's all over the place. This evolutionary nonsense. It's just stories. So question it, look into it, investigate it. And we just had Darwin's Day happen. And guess what? They didn't question anything. I was on social media challenging people. Question this. All you would get is hate. That's how crazy these people are. They've lost their minds. So what do we see? Creation versus evolution. We get evolutionary phylogenetic rates all over the board, all trying to prove deep time evolution right here, all trying to point at those dates. Then we get the absolute observable dates from pedigree mutation rates, and they all agree with creation timeline, rapidly fast. Isn't that amazing? What we observe matches what we believed the Bible to say, according to the genealogies and the timeline. What are the odds of that? They could have been anything. It could have been 50,000 years old. That would have been confusing for evolution, but that would have been devastating for us. But yet it doesn't match that. And nothing does. When we look in animals, we find the same thing. Nathaniel Jensen pulled up studies that were done on fruit flies and worms. And what is this thing? A water flea. The rates are fast. The mutation rate's rapid in all of these different things. They did a pedigree rate on chickens. It's the exact same thing as humans, 15 times faster than it should be. Well, what's going on here? I thought chickens were related to dinosaurs. What is their mutation rate going back only 6,000, 7,000 years? What's it doing? Oh, maybe it went into the bottleneck of Noah's flood and came out, and the mutation and the mitochondria was reset. Could it be possible? Of course it is. Then we get into these sweeping gene surveys that also countered any predictions that were ever done by Darwinism. 90% of all animal life, genetically speaking, the same age. They didn't expect to look over and see aquatic life and land life all exactly the same. Another unexpected find of the study was species have clear genetic boundaries. They have independent lines of ancestry going all the way down independently. There's no convergence on these common ancestors going back in time. And the absence of in-between species is something that also perplexed Darwin. Like, he looked in the fossil record and said, where are they? I don't see these intermediaries. They should be everywhere. There's hundreds of millions of years for them. Can't find them. And then they just say, oh, fossils are rare. Another excuse, right? This conclusion is very surprising, and I fought against it as hard as I could. They were literally fighting themselves in their own mind because it was so devastating to evolution. Of course, they have their own rescue devices, and of course, they'll never go to bed sleeping at night thinking that they weren't true. But look at this. 90% of all animal life appeared at the same time. This debunks much about what we know about today about the evolution of species. You're right, it does. It's because this whole concept is nonsense. It's not even true. And when we look at Neanderthal, for example, what do we find? What side of the bottleneck are they on? Well, look at this. Their genome in the CO1 gene matches ours identically. So they had to be on this side of the bottleneck, because if they were before the bottleneck and genetic diversity broke it apart, and well, this would not be nearly identical. Look, there's only 6 mutations separating us from them in this region. That's nothing. And then when we look at human beings and gorillas, why don't we converge with them going deep into the past? Look how close this is, genetic similarity. Well, the genetic similarity being this high is devastating with what you just learned about how fast mutation rates are. On the bottom, you can see all the differences from mutations separating gorilla. But at the top, you see all of the differences that are non-mutation that are separating us from the gorilla. Those are created differences. And that's how we know we don't converge on them going back into the past. That's why we see independent lines of ancestry for all of these different things in genetics and in the fossil record. They lose their mind when you say that, but that is exactly what this study showed, and that's exactly what we would expect and what we predicted, and it's what we found. Then we go to Nathaniel Jensen and his Y chromosome mutation rate. His study showed that about 4,500 years ago, using high coverage, there was actually 2 studies, but one in particular that he pulled the data out of, connected the phylogeny together to show who was related, and then made a mutation rate based off of that. He found that sometime around 4,500 years ago, Noah lived. So let's break this down and look at the study, shall we? This is the study you're going to be looking at right here. This is Carman et al. 2015. So what he did is he noticed that they hid something secretly in the additional filter area. And they hid it because the information was extremely harmful to what they actually wanted. And they won't release the data, by the way, and they just discard it. And so Nathaniel Jensen went in.
And he noticed that they only counted indels, because the goal of the study was just to connect family lineage and not make a mutation rate. So Jensen went in and counted up the mutations from the raw data, and therefore got his own mutation rate from the study, which used high coverage. And that is how he got such a fast mutation rate.
It matches the mitochondrial mutation rates, and it's not slow like they've been saying. So he compared them to population growth rates using that Carman et al. 2015 database, and noticed, like, wow, this really matches pretty good. There's about a 25% historical growth capture rate with that Y chromosome. So then he said, okay, let's back this up for just a minute and use all of the gamma, alpha, and epsilon roots, and they captured 90% of all of the population using the rate. Interesting, right? That's exactly what we'd expect from the human growth rate population growth curve, and it matched the mutation rate. And it didn't only do that, but it also matched known dates of the Amerindian population. So it matched other dates outside of the Y chromosome historical population growth rate curve. They're matching known historical dates of actual events that happened, and we know when they happened. And then we line them up with the Y chromosome from that study, and the growth curves match. So then I took this as the criticisms that came from to Nathaniel Jensen and how they're actually answered.
But before we go on, let's listen to what Dr. Dan, Creation Myths, has to say regarding this argument, and then we'll hear the answer from Nathaniel Jensen himself before I go on and give mine.
The other thing to note with the Jensen ones is one of the two that he used that had very high coverage, which is great. You want high coverage, 40 times coverage, give me that all day long. But one of the two papers that he used, they weren't looking at mutations. They were looking at structural differences, so like translocations. They weren't actually looking at mutations. And what he did is they created a phylogeny, the authors, and Jensen identified father-son pairs on the phylogeny. And then the branch lengths were, there was a key with like a scale bar for like this scale bar is X number of differences. And what he did is he blew up the figure really big on his screen and then counted the pixels on the scale bar and used that along the phylogeny to figure out how many differences there were in all the father-son pairs. The authors never actually reported that because they weren't doing sequence-specific stuff. They were looking at structural differences, and the authors didn't actually do like the error. Like there's an error rate when you do DNA sequencing that you have to account for. The authors didn't do that. So the amount of mutations they found was indistinguishable from the error rate in that study. So he showed 4, and then he rejects 2 of them for low coverage, and he bases traced on the other 2. And one of those didn't actually measure the point mutation rate. The authors created a phylogeny for that based on their sequence data, but they were only sequencing to use to find structural differences. They weren't actually looking for like single nucleotide polymorphisms. And so they didn't do the typical like error correcting that you would do to account for the sloppiness in sequencing processes. So the mutation rate that Jensen documents in that one is indistinguishable from the error rate of the DNA sequencing itself.
So I was thinking that Dan was talking about the mitochondria mutation rate this entire time, not the Y chromosome. That's why I said this.
That was on mitochondria, right?
No, that was the Y chromosome one. That was one of the 2 Y chromosome studies that Jensen used as the basis for trace.
Oh wow, I thought that was I thought that was the mitochondria one from a
no,
long time.
It was the Y chromosome, one of the 2.
This argument sounded very familiar, and then it dawned on me. We've covered this before. Matter of fact, there's a video all about it that was made 1 year ago, answered by Nathaniel Jensen specifically on the word for word what he said. But here's the kicker. It's not actually his argument. It was made by Herman Mays, not Dan. Dan is using it now.
So this is for you, Dr. Jensen. Herman Mays says most of the Y chromosome differences are errors and not mutations, and what Jensen is measuring here are errors between fathers and sons, not mutations. So he's asking you to repeat that.
So the mutation rate that Jensen documents in that one is indistinguishable from the error rate of the DNA sequencing itself.
I'm going to share my screen again here real fast because, okay. So this is one of the key tables from the paper. This will lead now. This is again sort of long backstory to this specific question. There were 2 studies. I give the references here, and you can find them in the paper if you want to look up the details, where they tried to measure father-sons, or in this case, this initial study, Xu et al. 2009, they had 2 Chinese men of known genealogical relationship. They had a common ancestor in the 1800s. They got the Y chromosomes of the 2 men, counted the number of differences, and translated this to a per-generation mutation rate. And the rate right here, these are numbers that are not necessarily comprehensible. The point is they got a slow rate of mutation that fit evolution. What they did, though, was use low-quality data. The technical term is coverage, which we can get into if people want to get into the technical data behind it. It's well established in the secular literature that low quality, low coverage will miss DNA differences. Another study from 2015 using about, I think it was 274 Icelandic pedigrees, and there were 750 or so Icelandic men, where, yes, we have this common ancestor in the 1700s or whatever the number is. Iceland is not a very large country in terms of population size, fairly detailed genealogical records. great place to do this kind of study. Once again, though, they used low quality, low coverage sampling, and they got, once again, a slow mutation rate. These two studies are the ones that I'm saying point towards a recent origin for humanity. It's one of the many lines of evidence pointing towards, I would argue, a recent time scale. This study was the one where they had about 31 either father-son pairs or brother-brother pairs. They had high coverage, aka high quality DNA sequence, and they found a rate that was 10 times faster. Now, I say they found a rate that was 10 times faster. This is the paper where they literally say in the methods, we found a rate that was too high, so we created a filter to pull out data that didn't match evolution. And I'm surprised they actually said that. Okay, Herman Mayes' comment is about this paper. And this paper was a study where it's exclusively focused on measuring mutation rates. It's not just the Y chromosome. This was 50 Danish trios, father, mother, offspring. And the primary focus of the paper is the rate at which DNA changes in the 99% of our DNA that we get from both parents. And they were meticulous in their methods. Again, it's high quality, high coverage. They show graphs where the rate of the father passing on mutations is higher than the rate of the mother, with older fathers, more mutations than younger fathers. Anyway, just almost no stone unturned. That's my point. It's explicitly a mutation rate study. They look with excruciating detail at the DNA inherited from both parents. They look at different types of DNA mutations. There's single letter changes. There are chunks of DNA that change where, let's say, the DNA is copied and for whatever reason, the cellular machinery skips 5 letters. It's a deletion or it's an insertion. There's these sorts of things that happen. Anyway, there's all sorts of DNA changes that occur. They look at these in great detail. They also say something about the Y chromosome. So they've just done this exhaustive analysis for 99% of our DNA. They look at the Y chromosome. They say we looked at the chunk changes, small insertions or deletions. Indels is what they're called. They give a rate for that, but silent on the single letter change for the Y chromosome, which is what these studies have been looking at. That's the main focus of almost all Y chromosome studies when you look at human history. The number of single letter differences swamps every other type of difference in terms of the sheer number of them. So number one here, then, this study of the Danish trios, there was about, I think, 17 father-son pairs in the 50 families. They don't say a word about the single letter change, which was this mysterious omission from the paper. Now, I'm reporting a rate here because they published a family tree based on the Y chromosomes with a scale bar. And so from that figure itself, you can extract from those data a mutation rate. So what Herman Mayes did then, so he, of course, recognizes this is way too fast for evolution. He emails the authors, says, hey, these creationists are using your data to argue for a young Earth timescale. And first of all, Herman Mayes won't share the details of their entire exchange. Number two, the authors, he admits, confirm, yes, indeed, there are 17 father-son pairs, and I found them. Thirdly, the authors say, we think that this is due to sequencing error. Now, if anyone's ever done science, anyone's tried to publish a scientific paper, I mean, this is Science 101. Nobody cares what anyone's opinion is. I mean, I had a colleague in graduate school who would say, he was talking about how he reads scientific papers, and pretty much every single scientific paper has 4 sections. Introduction, so setting up the question they're trying to solve. Materials and methods, which they say here's the lot number of the kit that we use to isolate the DNA, and here's the methodology so anyone can reproduce it. Then they have the results section, and then they have a discussion. And so he mostly seriously said to me, he says, the only thing I read in papers is the materials and methods. He said, materials and methods is what the people did. Results is what they want you to think they found. And discussion is unvarnished, rampant speculation, which he thought was worthless. So when people do science, if you look at scientific studies, all that matters is the data. Nobody cares what you think about it. It's an opinion. You can publish an opinion piece, but there's a big old banner that says opinion when you publish it. It's the data that matters. So number one, again, in the original paper, it's odd that some of the key data was left out. It slips through in one of their figures, and that's what I used to extract this number. Number two, Herman Mayes asks them, what do you make of this? This is their second chance to simply publish the data. And they don't. They give apparently whatever We don't know all that they said because Herman Mays won't share the extent of the email, his private correspondence. They simply say, we think it's sequencing errors. And number 3, then, Herman Mays apparently thinks this is sufficient, as if scientific debates are adjudicated by what someone says. That's pseudoscience, and he knows it. That's not how you adjudicate debates. Now, I can tell you, so here we are. We've gone through 2 rounds of Moretti et al. Not publishing the data. What are they hiding? I can tell you we have independent lines of evidence to be able to test this. There's multiple independent lines of evidence that I go through in the book and in my papers that show this mutation rate to be true. So the answer to the question, Herman Mays says that they're sequencing errors. His claim is based on someone's opinion. It's pseudoscience. Instead of actually testing. So number 1, they could resolve this by releasing the raw data, but they haven't. Number 2, he could do an experiment to try to evaluate this, which he doesn't. Number 3, I have done multiple experiments to resolve this directly, or I should say indirectly. There's all sorts of ways you can evaluate the actual rate of change, which I've done in subsequent papers and in this book. And really one way to evaluate it is to look for the signatures of human history all throughout our DNA. You can see it in the Y chromosome-based family tree. There's an echo of Noah, and on the lines of evidence go. It's a really remarkable case study in how the leading critics of creation science respond to a large body of data. They don't engage the testable predictions. I've given ways for people to falsify what I'm doing. There's predictions I make that you can go out and test and say, hey, your predictions didn't come true. It's wrong. Instead, they're using these, by their own definition, pseudoscientific methods to try to undermine it. And again, these are the leading voices in opposition, which I think is remarkable. They're basically doing what they've accused creationists of doing, relying on authority, saying certain things can't be questioned, not doing experiments, not looking for chinks in the established armor. There's another quote I can add here too, because he published some additional correspondence with them. One of the other things the author says is, well, take a look at some of the other studies out there and see, I don't think you can find evidence from the other studies out there that would support this fast rate of mutation. Some of the studies that they cite include this low quality study, also this study where they literally filtered out data in opposition. So even these own authors don't seem to be familiar with the Y chromosome mutation rate literature or the circularity built into some of these measurements. So he's asking the authors, they don't even seem to know what's going on in the field, which is remarkable to me. And they don't publish the data. That would be the simplest way to resolve this debate. Just give it to us in a transparent way so we can all see it. And they've had 2 chances to do it and they haven't. So There's my long-winded answer. And
the original argument comes from a critic named Herman Mays, who emailed the authors of the paper to get their rebuttal from them. They made a statement as though it was a fact, but gave no evidence. So of course, these critics took their reply and ran with it without fact-checking the raw data on their own. Surprise, surprise. After this, the original paper waxed eloquent about the mutations in every genetic compartment but the Y chromosome, i.e., they've had 2 chances to publicly release the data in support of the Y chromosome mutation rate claims. They haven't. And so what are they hiding? We all know what they're hiding. The fact that once again destroys their deep time and confirms the biblical model. We know that they're wrong, not primarily because they're hiding any type of the raw material and data, but because multiple independent lines of evidence confirm that this mutation rate is true. Remember, we just looked at all the mitochondrial evidence to show that it's true. Then we have population growth curves based on the Y chromosome tree that show it's true. Regional populations of growth curves based on mitochondrial that's true. I just showed you that one. Confirmation between the trees and known events from the history of civilization, which was the last one that I showed, last chart, the arrival of new mutations reaching fixation and the expansion of new haplogroups all at the same time in the Middle East, which matches the biblical timeline and place, and discoveries of Genesis 10 genealogies at the base of the tree that line up perfectly. What does he mean by that? Well, if we were to expect the Tower of Babel to be true, and we're going to be looking at the mutation rate that he obtained, which was 3 mutations per generations, and there's a total of 440 mutations total in human beings, that's only 146 generations ago. So if you use a generation time of 30 years, you get about the earliest date of Noah's flood being 4,380 years ago. And if you use a generation time of 35, it takes you back to about 5,100 years ago, about 200 years after the flood based on the Greek Septuagint. So it doesn't really matter. It lines up perfectly. And when you actually test them for the what the Bible says would have happened in the time of Peleg, when the population was split off from Babel. Guess what we land on? His generation time. Like, what are the odds of this? What, you can't just say this is all fluke. What do we see? The haplogroup explosions for the Y chromosome right out of the Fertile Crescent, right exactly where we would expect it to be. Why not out of Africa? They've been living there for 150,000 years, and yet no population explosion, no haplogroup explosion, no new mutations rising. It lines up with our story. That's what's going on here. So let's take a look at other things that would also agree and concur with it. Well, here we have writing systems. They arrive around 5,000 years ago at exactly the same time. Here we see the invention of writing began about 5,000 years ago in history. How about mathematics? Exactly the same time, about 5,000 years ago. It just arises out of nowhere. How about medicine? Medicine, boom, rises about 5,000 years ago. Exactly what we would see. How about the oldest trees? Trees don't even die. Some of them are immortal. They don't go back past 5,000 years. They don't do it. What's going on? Exactly what we would expect. How about civilizations? They pop up and arise about 5,000 years ago as well. How about genealogies? Unbroken chains of royalty, lineage going back to Noah himself in exactly the same amount of generations mentioned in Nathaniel's work. You can't make this up. When did mutations show up? Oh my gosh, look at that. Randomly, mutations just started to arise out of nowhere about 200 to 400 generations ago. Surprise, surprise. Exactly what we would expect about 5,000 years ago, right? Here's another one. Harmful protein coding mutations, a large rosy about 5,000 years ago in the past. Look, in addition, certain genes they learned harbored only young, more damaging mutations that surfaced less than 5,000 years ago. And so we see it again here. Scientists confirm that the human genome began to rapidly diversify not more than 5,000 years ago. Well, yet again, there it is. Helium and argon diffusion rates about 5,100 years ago is their average rate. What's going on? How about magnetic field decay? Oh, look at that. 5,000 years ago, something happened, changed it all. How about coral growth rates? About 4,200 years ago, could have made all of the coral reef. How about the, in Africa, Northern Africa, we get the Sahara Desert, which is the oldest desert in the world. And they're finding that about 4,000 years ago, some drought caused it probably. So that started. It was all green, about 4,000 years old now. How about the recorded span of all of human history? Find the exact same thing about 5,000 years ago. They put it on magazine covers 5,000 years ago. Things just started happening. We don't know why. And this is a paradox for them. It's called the sapient paradox. With brains like ours, why didn't prehistoric humans wait millennia to start making civilization? Think about it. We come out of a bottleneck 200,000 years ago, and we don't do anything for 195,000 years. We just walk around, right? I'm going to go over here. I'm going to go, oh, let's walk at that. Look at that bear. Let's go hunt the bear, right? And then all of a sudden, we're just inventing math, writing systems, calendars, agriculture, farming. Boom, we just explode. Let's build a friggin' huge pyramid. Yeah, let's rock and roll. Matter of fact, let's build pyramids worldwide. I never met them, but let's build a pyramid over here. That makes total sense. So how about phenotypic diversity? If evolution was true, you would expect the Africans who've been living in Africa and isolated because of the Sahara Desert, at least 200,000 years before we could even break out of there in the last 50,000, that the phenotypic diversity of Africans would be much more diverse than the other people groups. But that's not what we find, is it? We find low phenotypic diversity in primates and extremely high phenotypic diversity in human beings. And just think, these humans that you're looking at here are humans that are alive today. We've lost a lot of genetic diversity that we saw even in the past because Noah had a massive amount. And as times went on, a lot of the phenotypic diversity that was our super strong point, like our super strong jaw and our super strong brow ridge and our larger cranium and thicker bones, all of that just got destroyed over time because of how easy life became and how we were able to manipulate foods and make things soft and cook everything and make it just so much weaker. Our bones, everything gets all weaker and not able to tolerate it. So what's going on? Let's wrap this up here with a great statement. Darwinism is no longer just a scientific theory, but a basis of a worldview and an emergency religion for the many troubled souls who needed it. How eloquently put. right? We just find evidence that corroborates everything that we're talking about. And it just so happens to match so perfectly, these mutation rates, to all of those other things that I just blew through very, very quickly. What are the odds of that? I mean, think about the statistical probability of all of this just happening to line up. And they go, oh, Jensen just made it up. He just randomly made that mutation rate up. Oh, really? He just randomly made it up, but yet made predictions on it first, and then made other predictions that also just lined up perfectly. And yet that also matched other things outside of that perfectly. Yeah. You can believe whatever you want, but that's not reality. What we've seen is exactly what we would expect to see if the biblical model of ancestry is true. And you can take it or leave it. Doesn't really matter. We're here to present the facts and the evidence. If you want to believe in a fairy tale and just stick to myths that negate all of those things, and just believe in some hypothetical mysterious mutation rate that existed sometime in the past, feel free. That's fine. But don't tell me that's science. That's a SpongeBob absolute madness. And you need medication immediately. And until next time.
I hope you enjoyed this video. Please make sure to hit that like button. It really does help. This is Mattman. Until next time. Introducing the Biblical Coin, the first ever ministry-based cryptocurrency, the ultimate in security with locked liquidity and renounced ownership. The team is doxed, and you can find all the information you need in the links below.